Welcome to JAMA Plus AI Conversations. I'm Roy Perlis, Editor-in-Chief of JAMA Plus AI, and I'm pleased to welcome today's guest, Dr. Kenneth Kehl. Dr. Kehl is a medical oncologist and services researcher at Dana-Farber Cancer Institute. His recent study, Clinical Trial Notifications Triggered by Artificial Intelligence Detected Cancer Progression, a Randomized Trial, was published in JAMA Network Open, and it examines the impact of AI-driven notifications on clinical trial enrollment for cancer patients. Ken, thanks for joining us today.
Thanks so much for having me.
So before we get into the specifics of your study, can you give our audience a little bit of background on the broader issue? So, why is it challenging to enroll patients in oncology trials?
This is a really important challenge and has been for some time in terms of advancing novel cancer therapeutics. Historically, under 10% of patients with cancer who are adults have enrolled in clinical trials, and yet at the same time, clinical trials often do have trouble finding enough patients to answer their scientific questions. And I think some of the challenges include the fact that cancer treatment, of course, is increasingly personalized. So clinical trials may be for patients not just with a specific type of cancer or treatment history, but genomic biomarkers, for example, or other molecular biomarkers. So trials have gotten very granular and are looking for very specific subpopulations of patients, which sometimes make it challenging to find the right patient at the right time for that type of trial.
So naively, I would have thought that there'd be a huge demand and you would have people sort of banging down your door wanting to be in your trial. Has this always been a problem, or is this a more recent problem driven by, for example, the genomics and the fact that you're sort of slicing and dicing the populations, I guess, more finely?
Yeah, at a population level, it's been a challenge for quite some time, and that relates in part just to how adult cancer care operates in the US. Most adults with cancer are treated in community practice settings, which may have some access to clinical trials, but may not have the same level of access or same granularity that academic centers have. And that's really in contrast to, for example, pediatric cancer, where for decades, the large majority of children with cancer have been treated at tertiary centers and do participate in clinical trials. So it's partly structural, and then it has also, again, become more challenging as the subpopulations for trials are getting increasingly more specific.
And so right now, sort of before this kind of application, like how would you try to increase enrollment? What's the standard in the field? What do you do now?
The general sort of practice of clinical cancer research is, I would say, quite manual and human, and there are good aspects of that, of course. But for patients who are coming to a center where a trial might be open, it's up to the oncologist to be aware of what the trial options are, and that's true both in academic centers and in community practice. And sometimes patients do come in interested in a particular trial. They may have done their own research and they're acting as their own advocates. But of course, much of the time, a cancer diagnosis or need for a change in treatment is overwhelming. So it's really been up to the individual doc and patient in the past to try to find reasonable clinical trial options.
And before we get into the specifics of the trial, there's a model driving this ascertainment. What does the model look like? So what are you actually picking out of the imaging reports? Is that right?
That's right. So broadly, the goal of this work was to make the investment in our field in precision oncology, namely getting as much clinical and molecular data as possible about a patient and their tumor, more actionable, in part by trying to match patients to clinical trials. And one of the challenges in the past has been that we've had a tool developed by some great members of our team at Dana-Farber that can take in the genomic biomarkers of a patient's tumor and the list of biomarkers eligible for a particular trial and find trials that are potentially matches for the patient based on the genomics. But the problem is that a patient's disease evolves along their trajectory as they receive a treatment and may benefit, but then the cancer may become resistant. And so the goal of the AI model we developed was to identify what we had called clinical inflection points, namely moments in time when patients essentially needed a change in treatment, usually because their cancer was worsening. And so we did train a model to look at imaging reports, which for patients with solid tumors are usually the way that we assess response to treatment and progression of disease, and predict, based on each new report and the sequence of prior reports, whether the patient was likely to change treatment in the near future. And we trained that with our own retrospective data and validated its performance in an internal validation set. And that was the model that we were deploying in this study.
And it's operating over the radiology reports. So in this case, you're not parsing the images, you're just sort of riding along. And as the radiology report comes in, presumably you apply your model to that. And then what? What happens after that?
Right. So the model essentially generates a number, a prediction of the probability that the patient will change treatment, right, based on the sequence of CT scans or PET scans or MRI reports that have been generated for the patient. And then in the current study, we essentially ask the question, does delivering information about clinical trials when our AI model predicts that a patient will change treatment to treating oncologists increase the proportion of patients participating in clinical trials of new cancer treatments?
So what made you decide to do a randomized trial? Because I think an emerging topic in our field, I would say, is if you have something that is just sort of doing triage in a sense, is moving people around, I could imagine settings where it might have been tempting to just deploy it and see what happened. How did you decide to do a trial?
It's an important question. We had conducted a pilot of this process before moving on to a randomized study. And in that pilot, we essentially did sort of just deploy it, but with metrics we were collecting together with our phase 1 clinical trial group to try to understand, was the model doing what we thought it should, and sort of on a more anecdotal or single arm basis, were we getting patients on clinical trials more often? And, we knew for a fact from interacting with clinical researchers that this sometimes happened, but right, we wanted to understand whether that type of intervention at a more health system or institutional level made an objective impact, partly so that we can inform future efforts in this regard to try to make them as impactful as possible.
So this is a very unscientific observation, but I feel like rolling out a study like this would involve endless meetings, basically because it touches so many parts of the hospital, right? How was it getting from, okay, we have this technology that seems to work in a pilot study to actually rolling out an RCT that involves communicating with the docs?
It had some interesting elements. We did write it up as a formal research protocol in a place where there are hundreds of such protocols active. But it's an unusual type of study compared to a standard clinical trial, phase 2 trial of a new drug. There were meetings with our clinical research team and IRB around what actually is this study? Is it itself a clinical trial? And different folks actually have had different opinions on that. But we eventually did get it going. We had support from our clinical trials infrastructure team, which of course has as its mission trying to find patients who might be eligible for trials and helping trials accrue as efficiently as possible.
So in some ways, this is a best case scenario, right? Because you have buy-in from the institution at a place that has a culture of clinical trials. So it seems like you were well positioned to do this study. So it's interesting to know that it still involved some navigation.
I think we were well positioned to do the study. The trade-off, I think, maybe as we'll discuss in the results, that an academic tertiary care center may not be the kind of place where this kind of intervention is going to be most impactful. So made it easier to do, but I think may have impacted the effect we were able to have.
So let's go there. What did you find?
Our primary outcome in this study was whether patients with solid tumor cancers whose tumors had undergone sequencing through our institutional platform to try to drive clinical trial accrual and research were more likely to participate in therapeutic clinical trials if their oncologists were receiving notifications about genomically matched trials when our models predicted a treatment change or essentially progressive disease? And the answer was no. They were no more likely to participate in clinical trials.
Which was not what I expected. So I started reading this trial thinking, hey, this is really cool. I wonder how well it worked. And then came to the results and started scratching my head. I mean, what do you make of that?
Of course, I think anytime one runs a negative clinical trial or clinical study, one can construct plenty of potential explanations for why it was negative. I think, as I noted in this case, I think to some extent we were preaching to the choir. So we were sending information about clinical trials to oncologists practicing at an academic center, a tertiary care referral center, many of whom were quite familiar with the trials available in their disease centers. Sometimes we're not as familiar with the trials available in other disease centers. So if I treat lung cancer, I may not know what trials are available in colorectal cancer or in our phase 1 clinical trials group. And so we hoped we might have some impact there. But I think that certainly may have been a factor that made it harder for this type of intervention to have a quantifiable impact.
So as a trialist myself, I know we often feel a little sheepish when we have a negative trial, but this strikes me as a really important negative finding, or at least a really useful one, not least as a proof of concept. I mean, you do set the bar at, if you have a new tool like this, here's how you can do a trial. If you were to do it again, what would you do differently, if anything? Like, how would you, I don't want to say stack the deck, but what would you do to increase the likelihood that you see a difference?
Right. To some extent, we are already hoping to do it again, but this time focusing on a broader tool that can match patients to clinical trials, not just if they've had a broad genomic sequencing panel sent on their tumor, but just if they've been treated for cancer. And that history is recorded in the electronic health record, with the goal of piloting and testing that tool starting in our affiliated regional community practices at our cancer center, where the oncologists practice more general oncology. They don't just see lung cancer, they see colon cancer, breast cancer, et cetera. So I think that's part of how I might design this again. I think ideally it would be multi-institutional as well. So there might well be a different impact at a different center. And we are working on getting that kind of effort going as well.
Do you think there are lessons beyond oncology? Because we're sort of, I think for AI in general, we're kind of past the, isn't it cool that we can build these models stage? And now we really are in the clinical investigation stage. And as I said, one of the things I really like about your study is it's an effort to do a clinical investigation of an AI-driven intervention, even if the intervention is getting into a trial. Did you learn things that would be useful to other people who are trying to design these kinds of studies, even beyond oncology?
I think so. I mean, certainly the fact that we did the study demonstrates that it's possible to conduct a formal evaluation of a tool like this just in the same way we would of a drug or a medical device, though certainly this is more about surfacing information that a patient's physician might already have had access to if they sort of knew where to look. We weren't automating any clinical decisions, which I think is an important distinction. I think the more of these randomized evaluations we do, the better. I think AI is being increasingly incorporated into our day-to-day clinical practice. I know that for the last few months, I've had access to an AI scribe through our medical record system and have not seen data on how accurate it is. I know I have to correct it a fair amount, or what's the impact on either our workflow or on the quality of patient care. So I think you're right. We are beyond demonstrations of feasibility and moving into the need to treat this tool, much like we have other types of tools historically.
That's all the time we have today. Ken, thanks so much for talking to us today about your study in JAMA Network Open. To our listeners, if you want to read more about this study, you can find a link to the article in the episode description. To follow this and other JAMA Network podcasts, visit us online at jamanetworkaudio.com or search for JAMA Network wherever you get your podcasts. This episode was produced by Daniel Morrow at the JAMA Network. Thanks for joining us, and we'll see you next time.
This content is protected by copyright by the American Medical Association with all rights reserved, including those for text and data mining, AI training, and similar technologies.